Well, the modern science was founded by Gregor Mendel, where he changed biology forever. As you may know, pea flowers are normally self pollinated. This means that the sperm cells fertilize the egg cells from the same plant. What Gregor Mendel decided to do was cross his stocks of true breeding plants. He needed to cut the male parts from the flowers. Then he dusted the pollen from a different plant onto the female part of that flower. He got plants from two different parents with two different traits, and the result of this method is called hybrids. Well, Mendel's second conclusion is called the principle of dominance, where he states that there are some alleles that are recessive and others that are dominant. The delivery of characteristics from parents to offspring is called heredity. The study of heredity is known as genetics. The process where male and female reproductive cells join together to produce a new cell is called fertilization. A trait is a specific characteristic, such as seed color, height of a plant, of an individual. The factors that are passed from parents to offspring are called genes, and the different forms of these genes are called alleles.
So we're gonna get into the topic of probability and Punnett squares. Here we're gonna talk about Mendel's principles and Mendel's work with pea plants. Whenever Mendel performed a cross with pea plants, he categorized and counted the offspring. He had plenty of data to analyze. For example, whenever he crossed two plants that were hybrid for stem height, about 3/4 were tall and about 1/4 were short. That's when Mendel realized that the principles of probability could be used to explain the results of his genetic crosses. Organisms that have two identical alleles for a particular gene are said to be homozygous. On the other hand, organisms that have two different alleles for the same gene are heterozygous. As we can see in the diagram, an example, all the tall pea plants have the same phenotype or physical trait, however they don't have the same genotype. We can see that there are three different genotypes. The diagram that you just saw was a Punnett square, one of the best ways to predict the outcome of a genetic cross. The Punnett squares uses mathematical probability to help predict the phenotype and genotype combinations in genetic crosses.
The two-factor cross F1. So first, Mendel crossed true breeding plants that produce only round yellow peas with plants that produce wrinkled green peas. Plants were all heterozygous for both seed shape and seed color. These cross do not indicate whether genes assort or segregate independently. It provides the hybrid plants need to breed the F2 generation. In the second part of this experiment, Mendel crossed F1 plants to produce F2 offspring. For seed shape, segregate independently of those of seed color. Genes that segregate independently, such as the genes for seed shape and seed color in pea plants, do not influence each other's inheritance. The independent assortment helps account the genetic variations observed in plants, animals, and the organisms, even when they have the same parents.
When some alleles are neither dominant or recessive, they are called incomplete dominance. When both phenotypes are expressed, it is called codominance. Many genes exist in different forms, and it is called multiple alleles. And many traits are produced by the interaction of several genes. Polygenic traits are the traits that are controlled by two or more genes, and the environmental conditions can affect the gene expression and also the influence genetically determined traits.
Small disorder is when non-disjunction occurs in meiosis. For example, if two copies of an autosomal chromosome fail to separate during meiosis, an individual may be born with 3 copies of chromosome. This condition is known as trisomy, and the most common form of trisomy is the Down syndrome. Disjunction of the X chromosomes can lead to a disorder known as Turner syndrome. A female with Turner syndrome usually inherits only one X chromosome. Women with Turner syndrome are sterile, which means that they are unable to reproduce. In males, non-disjunction may cause Klinefelter syndrome, resulting from the inheritance of an extra X chromosome, which interferes with meiosis and usually prevents these individuals from reproducing.
Changes in a gene sequence can change proteins by altering their amino acid sequences. This can directly affect one's phenotype. Human genetic disorders can be caused by individual genes. Some examples are the sickle cell disease, the cystic fibrosis, and the Huntington's disease.